Welcome to The Explainer. So today we're going something really exciting. We're taking a highly structured, intense deep dive into the absolute most heavily tested concepts in the Neet biology exam, straight from your NCERT Class 11 and 12 textbooks. If you're grinding for this massive test, you already know the struggle, right? Bridging that gap between reading dense, dry textbook paragraphs and actually applying that knowledge to tricky multiple choice questions, well, that's the ultimate challenge. So today, we're focusing on exactly that, transforming standard textbook material into a high-yield blueprint so you can absolutely conquer test day.
Okay, let's dive into this. Consider this question for a moment. This is an actual past paper question that has literally tripped up thousands of students. It asks for the final proof that DNA is the genetic material. I'll just let that hang there for a second. Do you know it?
Yep, it's the experiments of Hershey and Chase. Knowing this rapid-fire fact, and specifically distinguishing it from the earlier work of Griffith or Avery, McLeod, and McCarty is our literal cheat code today. We are hunting for these exact golden rules that examiners absolutely love to test.
All right, here's our roadmap for today. First, Pillar 1, the molecular code. Then, Pillar 2, the botanical miracle. Next, Pillar 3, the engine of life. And we'll wrap it all up with this synthesis.
Jumping right into Pillar 1, the molecular code and unlocking DNA replication.
6.6 billion. Think about that number for a second. That represents the sheer scale of base pairs in typical mammalian and specifically human diploid DNA. All of this massive amount of information is tightly packaged into a space way, way smaller than a typical nucleus, roughly 10 to the power of -6 meters. But what's truly mind-blowing and honestly highly testable is how this massive code manages to copy itself.
Way back in 1953, when Watson and Crick proposed the double helical structure of DNA, they wrote this legendary line. It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism. It's just such a brilliant, understated realization, right? And it essentially birthed our entire modern understanding of DNA duplication. So the crucial point is this concept of semi-conservative replication. Basically, it means the two strands separate and each one acts as a template. So the resulting DNA molecule always has one old parental strand and one newly synthesized strand. And this wasn't just a wild guess. It was elegantly proven by Maselson and Stahl. They used heavy nitrogen isotopes to basically track the DNA over several generations.
I like to think of DNA replication as a tiny, highly efficient factory assembly line. The main machine running the whole show is the DNA-dependent DNA polymerase. And wow, it works incredibly fast. We're talking catalyzing around 2,000 base pairs per second in bacteria. But it's not just about speed, it requires a super high degree of accuracy, because obviously, mistakes lead to mutations. Now, pay attention here because this is a frequent exam target. Because the two strands of DNA are anti-parallel, replication is continuous on one strand, but discontinuous on the other. Those discontinuous pieces, we call them Okazaki fragments, and they elongate the lagging strand away from the replication fork.
Okay, let's see how you apply this concept. Take a look at this tricky question right from the 2017 Neet paper. In bacteria, when exactly does DNA replication occur? Does it happen during the S phase, like it does in eukaryotes? Actually, no. The answer is prior to fission. Bacteria simply don't have a defined S phase like human cells do. And it's these quick, specific facts that are the exact golden rules you're going to need for test day. Moving right along to Pillar 2, the botanical miracle. We're decoding double fertilization. Now, your textbook highlights double fertilization with this very specific phrase, an event unique to angiosperms. Pro tip. Whenever the textbook calls something unique, you absolutely need to mentally highlight it, because that is a massive critical identifier for exam questions. Let's walk step by step through this beautiful botanical mechanism. So first, after pollination, the pollen tube grows down through the tissues of the stigma and style. Second, it reaches the ovule and enters the embryo sac. Third, and this is key, the tube discharges not one, but two male gametes into one of the synergids. And finally, two entirely distinct fusions take place inside the embryo sac simultaneously. And this brilliantly illustrates what we actually mean by double fertilization. Let's break it down. On one side, we have syngamy. That's where one male gamete fuses with the egg cell to form a diploid zygote. On the other side, we have triple fusion. The second male gamete fuses with two polar nuclei in the central cell to produce a triploid primary endosperm nucleus. So
2 fusions happening at once equals double fertilization.
The sheer efficiency of this system is just incredible. The zygote goes on to develop into the embryo, which is the actual future plant. Meanwhile, that triploid nucleus develops into the endosperm. Think of the endosperm like a packed lunch. It provides essential nourishment to that developing embryo. So this tiny micro-mechanism perfectly dictates the macro result, which is a viable living seed.
All right, shifting gears to Pillar 3, the engine of life. Let's sprint through some high-yield human physiology.
72. This number represents the average beats per minute of the human cardiac cycle. And this steady pace, it's set by the sinoatrial node, or SAN. The SAN generates action potentials and essentially acts as the heart's own natural pacemaker.
Now, that beating heart drives what we call double circulation. It's truly an evolutionary marvel that we see in birds and mammals. We're talking about a closed system featuring 2 distinct pathways: the pulmonary circulation, which goes to the lungs, and the systemic circulation, heading out to the rest of the body. This strict separation is huge because it ensures that oxygenated and deoxygenated blood never, ever mix. It guarantees highly efficient oxygen delivery.
Now, what's really interesting about this slide is how we can take the incredibly massive topic of physiology and organize it into the super easy to memorize cheat sheet. You see, examiners love testing your knowledge of normal functions by asking you about disease states. So, for respiration, you need to focus on alveolar gas exchange and the pathology of emphysema. For circulation, zero in on the blood supply to the heart itself and coronary artery disease. And for excretion, look at urine characteristics and how, say, the presence of glucose is a major red flag indicating diabetes mellitus. Let's zoom in on that respiratory pathology for a second. Emphysema drastically reduces the alveolar surface area due to damage in the alveolar walls. I'm emphasizing this specific mechanism because it appeared exactly like this, literally word for word, in the AIMT 2015 paper. It's usually caused by cigarette smoking. Knowing the precise textbook mechanism of these diseases is completely non-negotiable if you want to succeed on this exam. Let's move to and see how this builds into our final section, connecting the biological dots, the synthesis. So throughout our explainer today, we actually started on the reductionist biology side, looking at the incredibly detailed physical, chemical, and molecular parts, right? Things like DNA replication and polymerases. But then we zoomed way out to systems biology, looking at the emergent properties of the whole, like double circulation and how the respiratory and circulatory systems constantly interact. To truly master biology and absolutely crush your exams, you have to be able to seamlessly zoom in and out between these 2 perspectives. Which leaves us with a pretty cool final thought to ponder. Can we truly understand the magic of life just by looking at its isolated molecules? Or does the truth only emerge when the whole system beats together? Keep asking yourself these kinds of questions, keep connecting those dots, and you'll find that cracking this subject isn't just about endlessly memorizing facts for a test. It's about genuinely understanding the very engine of life. Thanks so much for joining me on this explainer, and keep learning.